All right. Test, first page, not in front of the camera. It's exactly like this. Okay. Oh, not like that one. Like this one. Okay. I'm going to have 4, just like A, B, C, D, and I have 1, and I ask you questions. And basically, let me see if they're exact same questions. Yep, they're the same exact questions. So if you can do it for these, you can do it for the ones of the test. The whole front side is exactly what you see on this board here. Let's go over that in a second. Well, just real quickly, on the back side, I've got some pictures similar to what you've seen before on both the bacteria lab and in my notes. No. And they're going to be of the type of bacteria, morphology, prefix and suffix. It'll be very obvious. I know on the one on the next board, it only could see one of them, the spirillum, but you couldn't tell if it was diplo or staph or whatever. But you will be able to tell very easily on the test. Then underneath the bacteria questions, there'll be questions that are going to come right out of that worksheet. If you look in your notes right now for the polymer, or for the protein synthesis worksheet, look for that right now, because I'm going to tell you the questions are going to be just. I actually, this is a brand new test now. Remember. And I, in some cases, copied and pasted the exact question, or only tweaked it a very small amount. So if you could, you should look at that protein synthesis worksheet, which Dayton is working very hard to find right now, in your notes. And that sheet is going to be key. The blank questions, because I'm not going to ask you a question like describe from start to finish transcription and translation. That would be too hard for me to read and too long for you to write. I'll ask you specific questions like where does this take place, what does this produce, stuff like that. And by the way, again, there's a lot of little stupid mistakes we make along the way about this. I want to try to point those out, all right, as we go along. All right, and then by the way, underneath that, there's going to be a question like this one here. You're going to have a translation into messenger RNA and then into possibly even into the besides the amino acids, the phenotype. So for example, if there's a choice of genes, which one's going to be a square face, round face, hair, hairless, that kind of stuff, 3 feet, 2 feet. All right, I'll have to give you, again, the information that you need to solve that. Oh, and lastly, lipids. There'll be questions on lipids. So all of this, basically, this is a very good review. If you can do these questions, but the problem is going to be, can you do these questions without your notes? I mean, you have your notes in front of you. It's not that bad. All right, but let's take a look at this first page. Now, let me just give you a couple of hints. Okay, because what right now a lot of you did, and it's going to cost you points in the test for 2 reasons. One, you can't do it. You can't memorize every polymer in the world. That's not going to happen. So you look for similarities, number 1. Okay, and the second thing is you look for what the monomers are. All right, in this first case, this is the key. You should recognize a sugar molecule by now.
It says right there.
Okay, and it even says monosaccharide there, but it's not going to on a test. Okay, but you should recognize a glucose molecule, right? And so you put glucose molecules together, you know you're making a polysaccharide, which can either be, as far as we know, starch or cellulose, right? Which one is this?
Starch.
And you know that because they're all the same side, right? They're all the same way. They're not flipped every other one. All right. This guy right here. All right. This is one of the ones you'd have a little bit more memorization involved, but it's really not that hard. Say this were, for example, this word H here instead. It's pretty obvious that that's ethene, right? So what would that be if that were an H there? Polyethylene, right. The fact that it's a Cl there tells me it's what? Polyvinyl chloride. Okay, so you basically get, you don't have to memorize every single one individually. And I know that's going to cost you down here, I'll bet you, with people who look stuff up. But let's go to this one. Now the second way, besides knowing the monomer, the second way to know them is by what's happening at the what's happening here? What is happening at this junction where they're combining together? And what you'll see here is this guy's made of an amine part and an acid part, and they're joining together at that point. So if you've got a guy with an amine part and acid, it doesn't matter what's in the middle. I showed you this several different ways. I had different compounds or different graphics from the internet that I could have just had an R there, or I could have had whatever there. Either way, if it's got an amine on one side and an acid on the other side, it must be an amino acid. Now, here's a mistake I see. I saw it on the worksheet I gave you. You cannot call this as a product, for example, is not amino acids. What is the product called? What is the polymer called? I would accept polypeptide or protein. Polypeptide would be the more exact answer, but protein would be correct too. All right, so basically, don't say that the product of this is amino acids. It's not. The polymer isn't amino acids. The polymer is polypeptide or protein. Now, this is the guy that caused the most trouble. That's why I want to make sure you all tried this one, even though it was the last one. It's got a benzene ring in it, so people say, oh, where did I see a benzene ring before? Oh, it was in polyurethane. That's polyurethane. It's not polyurethane. Okay? Again, you have to look at what's happening. Like you looked at what's happening there. Look at what's happening here. You've got a carboxylic acid joining with an alcohol. Have we seen that before? Yep. Sure. We saw that under esters. That was the reaction for an ester. And since it's a dicarboxylic acid and a dialcohol, I'm going to have a polyester because it's going to keep growing either way. So that's what this guy is, polyester. This guy up here is polypeptides or proteins. All right, now let's talk about the other things you were asked about these guys. Clearly, the condensation ones are going to have water or some other molecule given off. So those are the three condensation ones, right? Yep. The other guy's addition. Let's talk about natural versus synthetic. Now, somebody, I can't believe it, you still have to ask, really, you should know that starch and cellulose are natural polymers. Amino acids, proteins are natural polymers. And you should also know that polyvinyl chloride, which is used in PVC piping, and polyester, which is used in your synthetic fibers for your clothes, are not natural, they're synthetic.
Maybe not PVC pipes just grow.
You don't have, they don't grow on trees. PVC pipes do not grow on trees. Nor does polyethylene, if this had been polyethylene, saran wrap doesn't grow. That, by the way, it goes for uses. Be careful of the uses. A use for PVC piping is not PVC. I get that all the time. Okay, you cannot say the use for PVC is PVC. The use could be piping. It could be PVC piping. It could be credit cards, but it can't be PVC. All right. And by the way, if I do ask for the name of the polymer, don't just write PVC. Write polyvinyl chloride. You can put PVC in parentheses. A use for starch or cellulose would be, a storage form of sugar. And you should know the difference between whether it's digestible or indigestible storage form. If it's starch, it's digestible, right? Yeah. If it's cellulose, it's indigestible. Okay. And then we've got the purpose of polyester is obviously easy. It's clothing. But this guy has, I guess this is kind of a tough question because there are so many purposes or uses for proteins. All right. And so basically, you could say enzymes, you could say pretty much anything that's protein. So again, that's kind of a tough one. Probably won't, if I do ask you for the use of this guy, it's because there are so many things. It does almost everything in your body. It's done by a protein. All right. So anyway, kind of like the same answer, like the answer is protein. That's what the name of the polymer is. That's what a polymer is. And it's also the use of it. Well, in a sense, yes, because proteins are what make you anyway. All right, so that's the only one that would be a little confusing as far as the use. All right, good enough. So you're going to whole front side is on polymers. We spent a lot of time on polymers. Okay, you need to look them over. And again, concentrate on identifying the monomers. That'll help. Identifying what's happening at those functional groups. You have a carboxylic acid and an amine. It's an amino acid. You have an alcohol and an acid. It's going to be an ester reaction. We've seen those before, polyester. Now, let's go to some bacteria questions. All right, well, we'll start with the DNA. How do we translate these? You got to be careful. When you go into the mRNA sequence, what's going to happen here? When I go from T, he's going to match up with what? A. I knew I'd do that. Okay. He's going to match up with A, but A matches up with what? U. Yeah, because... Way off. Wow. Good thing I'm not writing much.
Oh, it's like my handwriting.
Yeah, that's pretty bad.
It's like your actual handwriting.
A, U. And then the C matches up with what?
G.
G. Okay, so wow. Look at that. I got to read through the whole board again. I hate when it does this. Now, I'm not going to do all of these, obviously, but the big difference there is for the mRNA sequence, you're substituting uracil, right, instead of thymine, right? Then you look up on this sheet, and I'm going to give you this sheet. This sheet will... I'll make copies for everyone to have their own, and I made it bigger so it's not like the one on your notes is kind of small and hard to read, so it's a little bit easier to read this one. All right, and you'll translate those mRNA sequences into the codons that form whatever particular amino acid. All right, let's go to the morphology. That's pretty easy. This guy here, obviously two of them, circles, diplo, two, coccus, circle, or think of round sphere. So diplococcus. What would that guy be? With only that information, you could probably only say spirillum, right? You won't be able to say whether it's strep or whatever. And this guy, however, is clearly strep, right? Streptococcus. Okay.
Well, that's not clearly. Look at the top right. There's a huge clump.
Yeah, I know, but if you see a long string, I think you can tell that. And
what was the answer to B?
spirillum, just spirillum. Like I said, you're really not given enough information morphology-wise to tell what the prefix would have been.
I thought we always had that.
So what does staph mean, by the way? On the test, you will. On the test, the pictures I'll give you will be obvious. Staph means they're like clumps like grapes. And strep means string, basically think of a string. And diplo means two. The only one we didn't see on here was bacillus. That would be the shape of what? What kind of a shape? A rod shape? Okay, good. Now, describing transcription and translation, like I told you a second ago, definitely want to look over that sheet on protein synthesis. I could ask you questions like, on that sheet, where does this take place? What's the product of that? What are the names of the four nucleotide bases? What are they? Adenine, thymine, not codeine, guanine, and cytosine. All right, now, if in mRNA, it's uracil, okay? I could ask you, and you should be able to go through, because, I showed you all those video clips as well as the notes, and from start to finish, what's going on there, I'm going to ask you specific questions. I'm not going to ask you to draw or to give me an entire essay on it. Okay.
So I'm not going to have to actually...
No, I'm going to ask you specific questions like right out of that sheet. Where does this take place? What's the product of that? And what were some of the mistakes you made on that? I remember distinctly one of the questions asked you, what's the product of translation? And people put down amino acids, and that's wrong. The product of translation is not an amino acid. It's a polypeptide or a protein. Amino acids are what it uses to build that. Okay?
This is like the equation.
Right, exactly. Just kind of like I said there. All right, how about the lipids? We just did lipids last day, so it should be fresh in your mind. What makes up a lipid? What are the two big components to put a lipid together? Glycerol and a fatty acid. Okay, now here's the one that got people big time in the last period. What are the three functions of a lipid? What do I get? Give me some answers. Yell them out.
Cell membrane.
Okay, cell membrane, energy, and hormones. Okay, but for some reason, people just looked at the first thing they came across, and they wrote down underneath that fats, oils, triglycerides. They just wrote down whatever they saw under the thing first. Okay, that's not functions. Those are just examples of lipids. Cholesterol, fats, and oils are all examples of lipids. And finally down here, HDL, LDL. I want to explain that one more time because your class actually asked this question. I thought I made it clear, but I think I may redo the notes. This is the first time I did them. I purposely put in quotes, good and bad cholesterol. Okay, in reality, there's no such thing as bad cholesterol. And by the way, LDL and HDL are not cholesterol anyway. What are they?
Transport.
They're lipoproteins that transport, and they stand for high-density lipoproteins or low-density lipoproteins. And there's no such thing as good or bad, really. We do find a correlation between high levels of LDL, the bad cholesterol, all right, which again, it's not even cholesterol. We do find high levels of that correlating with heart disease, all right, but you do need it. You need cholesterol in every single cell in your body. Has to have it. You have to have it for the cell membrane. You need it to make your hormones. You need it to make vitamin D. You need it for a lot of stuff, all right, but there is the good and the bad. You should know which is which based on the fact that LDL does tend to build up. The oxidation products tend to build up in the arteries, causing plaque, atherosclerosis, possibly strokes, and heart attacks. Whereas the HDL's general function is to take those cholesterol out of the bloodstream and take it back to the liver where it's broken down. All right, so that basically covers everything for this test. You know exactly what to expect. All right, I don't think anybody's going to have